Welcome to Introductory Microbiology. Today what I'll be teaching you is the cultivation of microorganisms. And when we talk about cultivation of microorganisms, what do we mean? We are talking about the growth of microorganisms. You make conditions suitable for microorganisms to be able to grow and to reproduce. So that is how we mean by cultivation. In making these microorganisms conducive environment or conducive atmosphere for them to grow, microorganisms need water for them to grow. They need carbon source. They need, if they are aerobic, they need oxygen. If they are anaerobic, they don't need oxygen. And they need other things. They need nutrients. These nutrients come in various shapes and sizes depending on the exact type of microbes that we are interested in looking at. In growing microorganisms or in cultivating microorganisms, we have what is called a solid medium. We also can use the liquid medium. The liquid medium contains everything the microorganisms need for growth, but it's in a liquid state. The growth, you add everything it needs in a liquid state, so the microbes will grow, but it will be in solution. So that is what we call liquid medium, or sometimes we call it broth. In the case of a solid medium, it is the same liquid medium that we just add a solidifying agent called agar-agar to. This agar-agar makes it to become solid, and microbes grow on it, and they can be seen visibly on the petri dish if it is bacteria or fungi and other types of microbes. I'm going to give you the details of all this as we continue in the lecture. So we'll take a very short break and then we'll get back and settle down for the lecture in detail. This is just a summary. Now we are going to the lecture in detail. Come along and let us go. Cultivation of microorganisms. Cultivation of microorganisms, as I've said in introduction, is also the growth of microorganisms. Cultivation is the process of propagating organisms by providing the proper environment, type conditions. Parasites, bacteria, and viruses all generally require cultivation for the detailed study. The field of microbiology has the greatest experience in the cultivation of bacteria, and as such, bacteria divide by binary fission. This we have explained already. It also divides by asexual reproduction. And what happens during binary fission is that a single cell divides, giving rise to two cells. Those two cells rise to total of 4 cells, and they keep dividing from 4 to 8, from 8 to 16, from 16 to 32, from 32 to 64, from 64 to 128, and on it goes. This process of replication requires the acquisition of elements that make up their chemical composition. Nutrients from the environment provide these elements in metabolically accessible forms. In addition, organisms require metabolic energy to synthesize macromolecules and maintain essential chemical gradients across their membranes. Factors that must be controlled during growth include the nutrients, the pH, the temperature, aeration, salt concentration, and ionic strength of the medium. There are innumerable microorganisms, and therefore it is most essential to prepare a medium to study their morphological, biochemical, diagnostic, and other types of characteristics of basic and applied nature. Generally, any liquid or solid preparation made specifically for the growth, storage, or transportation of microorganisms or another type of cells is referred to as a growth medium or medium in microbiology. Most of the dry weight of microorganisms is organic matter containing the elements carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. In addition, inorganic ions such as potassium, sodium, iron, magnesium, calcium, and chloride are required to facilitate enzymatic catalysis and to maintain chemical gradients across the cell membrane. For the most part, the organic matter is in macromolecules formed by the introduction of anhydride bonds between building blocks. Synthesis of the anhydride bonds requires chemical energy, which is provided by the two phosphodiester bonds in adenosine triphosphate. Additional energy required to maintain a relatively constant cytoplasmic composition during growth is a range of extracellular chemical environments is derived from the proton motive force. So one energy, the main energy is from ATP, another energy is from proton motive force. The proton motive force is the potential energy that can be derived by passage of a proton across a membrane. In eukaryotic cells or eukaryotes, the membrane may be part of the mitochondrion or the chloroplast. In prokaryotes, the membrane is the cytoplasmic membrane of the cell. The proton motive force is an electrochemical gradient with two components, a difference in pH, hydrogen ion concentration, and a difference in ionic charge. The charge on the outside of the bacterial membrane is more positive than the charge on the inside, and the difference in charge contributes to the free energy released when a proton enters the cytoplasm from outside the membrane. The metabolic processes that generate the proton motive force are discussed. The free energy may be used to move the cell to maintain ionic or molecular gradients across the membrane to synthesize anhydride bonds in ATP or for a combination of these purposes. Alternatively, cells given a source of ATP may use its anhydride bond energy to create the proton motive force that in turn may be used to move the cell and to maintain chemical gradients. The growth media may be used for initiation of a culture or a subculture for enrichment, identification, or diagnostic tests. There are special media for the growth and maintenance of mammalian cells, plant cells, protoplasts, and other types of cells in tissue culture, such as for viruses. There are many chemoautotrophic bacteria which can be grown in simple aqueous media containing mainly or only a mixture of inorganic salts. Nutritionally simple, undemanding heterotrophs can be cultured on a large range of culture media as nutrient broth and peptone water. But nutritionally fastidious heterotrophs can be grown only on an enriched medium. When we said an organism is nutritionally fastidious, that means the organism will not just take any kind of food. It means rich food. It's like human being that need protein, beans, egg, vegetable, very rich food, not gari, eba, fufu. So in microorganisms too, there are some organisms that if you give them gari, eba, fufu, they will not grow. I'm just using that as example. Microorganisms don't eat gari, eba, fufu. But what I'm saying is that like some human beings, they need fish. They need fruits, they need meat, they need beans, they need protein,
The same way, some microorganisms are nutritionally fastidious, that just any food that normally will enhance the growth of other microorganisms will not enhance their own growth. They need to be given special food. So such organisms are said to be nutritionally fastidious. Nutritionally fastidious heterotrophs can be grown only on an enriched medium. You can see, because they are nutritionally fastidious, that's why you use enriched medium for growing them. You don't use a normal medium, you use enriched medium. You enrich the medium with nutrients or with factors that will enhance their growth. Strict anaerobes are often cultured on pre-reduced media. That means media poised at or below a particular redox potential. Oxidation reduction potential represented by E with the subscript. The poison agents used in the media are ascorbic acid, cysteine, hydrochloride, and thioglycolate. Poisoning is analogous to buffering in the context of pH. That is, E subscript H produced depends on the ratio oxidized reduced poison agent. The stability of E subscript N is influenced by the absolute amount of poison agent in the medium. The solid media are prepared from liquid media that have been solidified or gelled with an agent such as agar or gelatin. I've mentioned agar previously in my introduction. The medium which without supplement can support the growth of nutritionally demanding species of bacteria is often referred to as basal medium. So take note of the meaning of basal medium. I am going to ask you that in the exam. The medium which without supplement can support the growth of nutritionally undemanding species of bacteria is often referred to as basal medium. So I will ask you, what is basal medium? Define what you have to explain basal medium. The medium in which all the ingredients, including trace substances, are known quantitatively is called defined medium. I'm going to ask you to define defined medium too. The solid medium on which different types of organisms can be distinguished by their different forms of growth is termed differential medium. I'm going to ask to define differential medium too. A medium which contains substances that encourage growth of the required organisms and or inhibit the growth of other types of organisms is called enrichment medium. I am going to ask you to define enrichment medium too, e.g. selenite broth and tetrahonate broth. So look for the examples of the different types of medium. I am going to ask you to give examples too. The medium for the initial growth and the subsequent storage under conditions of minimal growth is known as maintenance medium. I am going to ask to define maintenance medium too. The maintenance medium for microorganisms is used to prepare a culture of the given organisms that can be stored either at ambient temperature or under refrigeration. Its subcultures are required at intervals ranging from 1 to 12 months. The constituents of the maintenance medium are often of minimum consistence with the need to maintain viability of the organisms that is being cultured, e.g. cooked meat, medium, and dorsal egg. The common media for cultivation of bacteria are required in a laboratory. The common media for cultivation of bacteria often required in the laboratory are chemically defined media. That's number one. What do we mean by chemically defined medium? A chemically defined medium is a growth medium or suitable for the in vitro cell culture of human or animal cells, in which all of the chemical compounds are known. Is chemically defined, so you know all the chemical compounds are known. It is used to grow chemoautotrophs and photoautotrophs and for microbiological assays. What do you mean by complex media? That's another type of media. Complex media are rich in nutrients that contain water-soluble extracts of plant or animal tissue, e.g., enzymatically digested animal proteins such as peptone and tryptone. Usually, a sugar, often glucose, is added to serve as the main carbon and energy source. It is used to grow most chemoheterotrophic organisms. Anaerobic growth media, commonly used anaerobic culture media, are Robertson's cooked meat broth, anaerobic blood agar, egg yolk agar, Bacteroides, bile, esculin agar, used to grow obligate anaerobes. Obligate anaerobes are anaerobic organisms that will not grow if a little oxygen is present. They need obligate. Obligate means a must. The condition for their growth must be anaerobic. Selective media, media that inhibit the growth of unwanted microorganisms and support the growth of the organism of interest, and they are called selective media. So this media, we allow some to grow, we disallow some from growing. Selective medium contain particular ingredients that inhibit the growth of certain microbes. An example of a selective medium is McConkey agar. It is used to suppress unwanted microbes and encourage desired microorganisms. It allows the growth of coliforms and does not allow the growth of non-coliforms principally. We also have EMB agar called Leucine methylene blue agar. This allows the growth of fecal coliforms like E. coli, but it does not allow the growth of other such microorganisms. Differential media. What does differential media do? Differential media contain compounds that allow groups of microorganisms to be visually distinguished by the appearance of the colony or the surrounding media, usually on the basis of some biochemical difference between the two groups. These are used to distinguish colonies of the desired microorganisms from others. So if you want to see the colony of E. coli, what you do is you use EMB. E. coli have a distinct color when it grows on EMB agar, and the color is dark greenish with metallic sheen. So if E. coli is present, it will show that color, while others will not show that color, and in fact it will not even allow the growth of others at all. The next media we'll look at is enrichment media. Enrichment media refers to the liquid media that inhibits the growth of unwanted bacteria. Enriched media allow the growth of a wide variety of microorganisms. Enrichment media allow the growth of a particular type of microorganism in the medium. The enriched media facilitate the growth of fastidious microorganisms. I've said this before. Enriched media facilitate the growth of fastidious microorganisms. I've explained what fastidious microorganisms are previously, and I told you that fastidious microorganisms are microorganisms that require special media or special conditions for growth. They don't just grow on any media like other organisms. So these are similar to selective media, but are designed to increase the number of desired microorganisms to a detectable level. So for now, this is where we're going to stop on the matter of cultivation of microorganisms. So there are different reasons why we cultivate microorganisms. I've told you about generation time, also called doubling time, which is the time it takes for a bacterium to reproduce. And let me just make it known to you that as we're talking about media, these organisms we put, for example, a differential media or selective media or basal media, it will be prepared and poured into petri dish with agar-agar added, or if it has come with agar-agar from the manufacturer, you prepare it, to pour into the petri dish when it is still hot after preparing the autoclave, and then what happens in the petri dish is that it will solidify. It will solidify when it gets cold. Now, when organisms are on it, if such plate is exposed to air, or the pour plate method is used, and then organisms are grown on the radial or stick plate, or any other type of inoculation is done, what happens is that each of the microorganisms, generation time for bacteria usually 20 minutes or less. So if generation time for a bacterium, let's say, is 5 minutes or 20 minutes, in 20 minutes, one single bacterium has become 2. In another 20 minutes, has become 4. In another 20 minutes, has become 8, and on. You remember we mentioned this in the class. And by the time it becomes 8, becomes 16, become all that, and becomes millions within even 10 hours or to 12 hours. So usually in 24 hours, that's a single day, the bacterium will have even grown to millions of millions, such that now what is invisible, usually as microbiology is studying invisible organisms, what is invisible, because it's millions and millions and millions growing on the same spot, it now becomes visible. I think you understand. It now becomes visible, and then we are able to see it and be able to you're able to work with it and use it for other beneficial uses. So this is the principle behind the cultivation of microorganisms. I'm going to talk to you some other time. Bye for now. On the cultivation of microorganisms, this is where we're going to stop the class for now. I have other materials on sterilization, on disinfection, and on other subjects in microbiology. I want to check them out in the playlist on microbiology. There are other videos that will benefit you on success and entrepreneurship too. I want you to subscribe to this channel and also to share my videos with other people. Also subscribe, and you are going to be glad you did. I'm going to talk to you some other time. Bye for now.